Our first lesson is all about the parts and functions of the microscope, while the second lesson is all about the levels of biological organization. So at the end of the lesson, the first lesson, you are expected to first identify the three main parts of a compound microscope and to describe the functions of each part of the microscope. So are you all ready to explore the majestic and interesting world of science? Let us start with our first lesson. So to begin with, let us first define a microscope. So a microscope is a tool which can help you see tiny objects and living organisms. So I hope most of you have already seen a microscope. So you can see a microscope in a laboratory or in a science laboratory. So the science of investigating small objects using such an instrument is called microscopy. Microscopic means invisible to the eye unless aided by a microscope. So a microscope is divided into three major parts based on their functions. First is for magnifying, second one is for illuminating, and the third one is the mechanical parts. So let us first have the mechanical parts. So when we say mechanical parts, it is the part of a microscope that is used to support and adjust other parts. So under these mechanical parts are the base, body tube, coarse adjustment knob, fine adjustment knob, inclination joint, stage, and stage clip. So let us know the different functions of these parts. First one is the base. Base provides support to the microscope. So if you have seen a microscope, the base serves as the fit of the microscope. It provides supports to the whole body of the microscope. So the second one is the body tube. It is a tube that separates the objective and the eyepiece and assumes continuous alignment of the optics. Third one is the coarse adjustment knob or coarse adjustment knob. So its functions is to allow the movement of the tube upwards or downwards. So through the coarse adjustment knob, we can move the body tube of the microscope upward or downward. The next one is the fine adjustment knob. It is used to bring the specimen into sharp focus under low power and is used for all focusing when using high power lenses. So fine adjustment knob is used to move the objective lenses to different power lenses. So next one is the inclination joint. So it connects the arm to the base. It helps incline the microscope for a more comfortable viewing. So inclination joint, its function is to connect the arm of the microscope to the base. So another one is that it helps us to, through the inclination joint, we can incline the microscope for us to be comfortable when viewing. Okay, next is the stage. So it is a flat surface where the mounted slide is placed. So from the word itself na, stage. So it is a flat surface where the slide which has the specimen to be observed is placed. And the last one is the stage clip. Stage clip, it holds the glass slide in place. So stage clip is used to avoid movement of the specimen observed. So it holds the glass slide in place. So the second major parts of the microscope is the illuminating parts. So the illuminating parts of the microscope, which should supply light and regulate light towards the focused specimen. So when we say from the word itself, illuminate. So illuminating parts is used to supply light and regulate light towards the focused specimen. So under this illuminating are diaphragm, stage condenser, and the mirror. So let us know the function of this part. So first one we have diaphragm. Diaphragm controls the light going through the aperture. So the diaphragm, which is under the stage, we can see the diaphragm under the stage of the microscope. So its function is to control the light going through the aperture. Next one is the stage condenser. So it is used to collect and focus the light from the illuminator onto the specimen. It is located under the stage, often in conjunction with an iris diaphragm. So stage condenser is also found in the under the stage. So its function again is to collect and focus the light from the illuminator onto the specimen. And the third one is the mirror. So I know you all know what a mirror is. So a mirror allows the light to pass through the hole in the stage and illuminate the specimen. So the function of the mirror in a microscope is, of course, to allow light to pass through the hole in the stage and illuminate the specimen. So then the third division is the magnifying parts. So the part of the microscope that specimen in focus appears larger. So from the word itself, magnifying. So it helps the specimen to magnify or to be viewed more clearly. So under these magnifying parts is the eyepiece and the objective lenses. So eyepiece, the function of the eyepiece is, it is a lens that is attached to a draw tube, or eyepiece is also known as the ocular. So some scientists also call this as ocular. Next one is the objective lenses. So objective lenses, it is responsible in holding the objective lenses such as the high power objective, the scanner, the low power objective, and the oil immersion lenses. So it facilitates the changing of objectives. So let us now discuss the different objective lenses that I have mentioned, which is the scanner, the low power objective, the high power objective, and the oil immersion. So a scanning objective lens that magnifies 4 times is the shortest objective and is useful for getting a general overview of a slide. So if you have seen a microscope, the short objective lens is what you call the scanning objective lens. So it is used to view the specimen in a general. It is used to get a general view of the specimen. So the next one is the low power objective lens. So a low power objective lens magnifies 10 times. But remember that it is coupled with an eyepiece lens. So the total magnification is 10 times the power objective lens. So if you, as we can observe, so if the scanning objective lens is 4 times, then a low power objective lens, low power objective lens magnifies 10 times. So that means that we can view more clearly the specimen through the low power objective lens. So the next one is the high power objective lens. So a high power objective lens magnifies 40 times with a total magnification of 400 times if the eyepiece lens is 10 times power, and it is ideal for observing very fine detail such as nerve cells in the retina or the striations in skeletal muscles. And the last one is the all immersion lens. So the longest objective lens is an all immersion objective lens, which magnifies 100 times. The total magnification is 1,000 times if the eyepiece lens is 10 times power. the detail of individual cells such as red blood cells. This lens requires a special oil to form a link between the edge of the objective and the cover slip. So, just a reminder, before you use an oil immersion objective lens, ensure the specimen is in focus under the high power objective lens. After you remove the high power objective, put a tiny amount of oil onto the cover slip above the specimen and then move the oil immersion lens into the position. So, that is the three divisions of the parts of the microscope, which is the mechanical parts, illuminating parts, and the magnifying parts. So let us now move on with the second lesson, which is all about the levels of biological organization. So biology is defined as the branch of science which deals with the study of life or of all the organisms on Earth. It is coined from the two Greek words, which is bios, meaning life, and logos, which means study of. So, biology, the word biology comes from the two Greek words, which is bios and logos. So, the word bios means life, while logos means study of. Okay, so all living things exhibit a unique and complex hierarchical organization. This is arranged in ascending order or from lowest to the highest level that includes cells, tissues, organs, organ systems, organism, population, community, ecosystem, and biosphere. One concrete example of a living thing is you or us humans. You are composed of many parts, and each part has a specific function. Every part is interrelated and is necessary for your continuous existence on this planet. It is guided by the rule, all for one, one for all. Because these parts of your body will not work alone. Instead, it works as a whole. One part will not be able to work properly if other part is missing. So after going through this lesson, you are expected to describe the different levels of biological organizations from the cell to biosphere. So specifically, you are expected to first classify organisms into their level of biological organizations and create a multiple pyramid of the levels of biological organization. So let us now discuss the different levels of biological organization. So organisms are categorized according to the number of cells in the body, either unicellular or multicellular. So, organisms is divided according to the number of cells in our body. So, it is either unicellular or multicellular. So, when we say unicellular, a unicellular organism depends upon just one cell for all of its functions, while a multicellular organism has cells specialized to perform different functions that collectively support the organism. As they grow and develop, they become highly organized and specialized. So,